everyone, welcome back to Windows Seat Studies. I hope you all are doing well. So we have been learning about histone modifications, in which we studied about histone acetylation as well as histone methylation. So let's continue learning about H3K9 methylation mark. So we learned in the previous lecture that this is a repressive mark, which is added or written by the enzyme SUV39H1. This is the writer enzyme for the methylation mark. Means this is the enzyme which methylates the H3 protein and the ninth lysine residue. So alongside the SUV39H1, there are other enzymes as well which can add this methylation mark. So we will learn about those other enzymes and what are the differences between that enzyme and the SUV39H1 as well. So, to do a quick recap, what we learned about in the last lecture. So, this methylation mark is added by SUV39H1, and this SUV39H1 adds the methyl group on H3 protein. So, after addition of methyl group, this provides a docking site for a protein known as HP1, heterochromatin protein 1, and this protein is responsible for heterochromatinization. So in the previous lectures we have learned about the mechanisms, how the HP1 is responsible for heterochromatinization. So HP1 recruits HDACs, histone deacetylases. So histone acetylation is active mark. So they remove that active mark, which leads to the repression of the genes. So that is why recruitment of histone deacetylases leads to the heterochromatinization. So this is how this is the mechanism of the heterochromatinization. So first HUV39 comes, it methylates the H3 protein at ninth lysine residue, and this provides a docking site for HP1. So the binding of HP1 to the H3 protein, this recruits the HDACs, the histone deacetylases, and this is how the active chromatin is converted into heterochromatin. So this is the whole mechanism. This is the whole flow of the process. So we learned about this in last lecture. So this is how H3K9 methylation is the repressive mark. But this whole mechanism, this happens at the region known as pericentromeric region. Okay, the region near the centromeres is known as pericentromeric region. So now let's learn about other enzymes, other writer enzymes which add the methylation mark. So the first one was SUV39H1. Okay. So the other enzyme is SETDB1. Okay. So the SUV39H1, this enzyme adds the methyl marks at pericentromeric regions. Okay. The regions near the centromeres. So the centromeres, the pericentromeric regions, the telomeric region, or transposable elements, repeat regions, all of those regions need to be silenced for maintaining the genomic stability. Okay, so we already know about it. That is why at the pericentromeric regions, the repression is done by the methylation, but that methylation mark is added by SUV39H1 enzyme. But the SETDB1 enzyme adds the methylation mark at transposable elements. Okay, so this is the difference between those two enzymes. But alongside this difference, there is those enzymes differ in the mechanisms also. So the SUV39H1 adds the methyl mark, and which helps in the recruitment of HP1 protein recruitment. So this serves as a recruitment signal. Okay, this serves as a recruitment signal for HP1. But SETDB1, but the methylation by SETDB1 is a bit different. So what happens is that there's a repressor known as CAP1. Okay, so this is a co-repressor. Means along with something, along with the main repressor, it represses a gene. Okay, so what happens is that CAP1 is KRAB associated protein 1. So this cap1 binds to the KRAB ZFP, means KRAB zinc finger proteins. So those proteins have zinc finger domain. Okay, so this zinc finger domain is the DNA binding domain. So this means that so what happens is that this KRAB ZFPs, means this KRAB proteins, they are DNA binding proteins. So these are the transcription factors, DNA binding transcription factors. Okay, so they bind to they bind to the DNA. Okay, so this is the DNA, and this is the KRAB ZFP, so KRAB proteins. So they bind to the DNA, and to which there associates the KAP1. Okay, so this is KAP1, the KRAB associated proteins, while this is the KRAB ZFP. Okay, KRAB zinc finger proteins. So this association, this association provides a scaffold for this provides scaffold for the enzyme SETDB1. Okay, so due to the recruitment of CAP1 to KRAB, which has been recruited to DNA, so it provides a recruitment signal, and it provides a docking site for SETDB1. So to which, so this to which, to this set, to this, okay, so to this SETDB1, this enzyme binds to this scaffold, and the binding of SETDB1 results into the methylation of H3 at ninth lysine residue. So this results in H3K9 methylation. So it can result in either a trimethylation, dimethylation, or monomethylation. Okay, so SETDB1 methylates the histone protein, and this methylation now will act as the recruitment. It will act as a recruitment signal for HP1, and for the so this will recruit HDAX, and now this will convert the active chromatin to heterochromatin. So this is how the SETDB1 mediated methylation is done. So alongside the KAP1, SETDB1 works, okay, and it methylates the ninth lysine residue. Okay, so it adds the methylation mark, and now this represses the genes or the transposable elements which are the represses the downstream genes. Okay, so this represses the genes which are downstream to the DNA sequence to which the KRAB ZFP binds. So this is how the both of those differ. So SUV39H1 can it can bind to the, so it can it methylates the H3 protein, and due to the methylation it recruits HP1. But for SETDB1 to act, it needs a scaffold of KAP1 and KRAB proteins. And the it adds methylation mark, which then recruits HP1, and it then heterochromatinizes the active chromatin. So this is the difference between those two enzymes. Uh, so this was in the different locus. Okay, in the SUV39H1 it does that methylation at pericentromeric region, while the other one ZDB1 it does it at the transposable element. But at telomeres there's something different happening. Okay, so again telomeres need to be silenced for maintaining genomic stability. So at telomeres, instead of HP1 there is recruitment of other proteins which are homologs of HP1, and those are known as CBX1 and CBX3 and CBX5. So these proteins are HP1 homologs. Okay, Okay, and so these proteins are recruited over the chromatin and they does the heterochromatinization. So this is the only difference at the telomeres. So this is how H3K9 methylation mark results in heterochromatinization, and that is why H3K9 mark is is a hallmark of heterochromatinization. Okay, so this is how H3K9 mark is a repressive mark. Okay, so the take home messages are, so let's summarize quickly everything. So which the important thing that you need to remember is H3K9 methylation mark is a repressive mark. Okay, it is the hallmark of heterochromatinization. It can be added by the so it can be added by the different enzymes. So this so the enzymes which add this mark are known as writer enzymes. Okay, so the writer enzymes for this marks are SUV39H1, SETDB1. Okay, so SUV39H1 this adds at pericentrometic region, while SETDB1 adds it at the transposable elements. There is other there are other enzymes like G9A, but you don't need to really focus on that for now. So SUV39H1 and SETDB1 are the two main enzymes that we learned about it. So what they do? They ultimately recruit proteins which are known as HP1. Okay, and those are they are responsible for heterochromatinization. So this is all that you have to remember for H3K9 methylation mark. In the next video we will learn about H3K27 methylation mark as well as H3K20 methylation mark. And then after learning about those methylation mark, we will learn about other histone modifications such as histone phosphorylation, histone ubiquitination, and histone tail clipping. Till then, see you. Bye bye.